Welcome everyone, this is Rabia Manzoor and we are discussing sampling techniques and sampling distributions. The topic of today's lecture are some more important concepts uh, that are important for sampling. That includes true value, what is true value, what is accuracy, and what is precision. Then in further this lecture we will talk about what kind of error we generally deal in sampling. So let's start with the important concepts: true value, accuracy and precision. Before explaining it, I am starting reading it through your textbook. True value. By true value, we mean the value that would be obtained if no errors were made in any way in obtaining the information or computing the characteristic of the population. So, true value of the population is possibly obtained only if the exact procedures are used for collecting the correct data. Okay, let's talk about the true value. Suppose we have a population and we are not performing any sampling, we are not taking a sample, we are studying the complete population. We are studying each and every element of this population. Then the numerical values which we will calculate by using the whole population, like its mean, its variance, its standard deviation, or any other numerical measurement which is calculated from the whole population without any error. This this value, numerical value, is called true value. So you can say true value is basically the numerical value which we calculate from the whole population, and we are not dealing with the sample. We are discussing a complete population, and from that complete population any numerical value which we obtain is called true value. Now, what is accuracy? Okay, accuracy. By accuracy, we refer to the difference between the sample result and the true value. Now, what is accuracy? Accuracy is basically the difference between the sample result and the population result. The smaller the difference, the greater will be the accuracy. Accuracy can be increased by eliminating the technical errors. Obviously, if we remove technical errors from the sample, then the sample results and the population results will be closer. And as will be closer, there will be small difference. So it can easily be obtained by eliminating the technical errors. Point number two, by increasing the sample size. Obviously, if we increase the sample size, if we have a population of 10,000 units and we are taking a sample of 30 units, so you can say 30 is very small in this case. And if we increase the sample size, for example, 100, 150, or 300. So 300, by 300 we mean we obtain more accurate results as we increase the sample size. So the sample results and the population results will be closer by increasing the sample size, and it will result in accuracy. Now let's talk about accuracy by using this example. Suppose we have a population and we have studied all the population, so we will get our true value from this population. And now after calculating the true value, we are taking a sample. Okay, we are taking a sample, a small information from the population, and we will calculate the same measurement from this sample too. For example, if we studied population and we calculated the mean. Now, by using this sample, we also calculated its mean. So the difference between the sample mean and population mean will be smaller the difference, the accurate the results will be. So it's not the difference basically. If the accuracy is basically the sample information minus the true value, the smaller this value, the more accurate your sample results will be. Now, The next concept is what is precision. So let's talk about it again. Precision, by precision we refer to how closely we can reproduce from a sample the results which would be obtained if a complete count census was taken using the same method of measurement. So precision is a little, you know, it's kind of a concept which tell you how close the samples or the reproduction is. Like what? From a population, we do not obtain a single sample. We draw more than one sample. So the results we obtain from this sample, the closer the results would be, the more precise your uh, work, your research will be. So by precision, we mean the same. How can we reproduce the same results from the repeated samples? That means the precision. Let's I clear it through a simple example of dartboard. I have four dartboards. At this picture, you can see this is the center and this is the mark, right? So these are your marks. The results of each mark. You can see there are 1, 2, 3, 4, 5, 6, 6 samples, different samples or different shots in this case. There are 6 different shots and the shots are very closer to each other. They are not accurate because they are far different or they are far from the actual point, accurate point. They are far from the actual point. That means that they are not accurate but they are precise. There is precision because the results are repeated at a same point. You are getting same kind of results, right? That's why they are precise but they are not accurate. In second figure, you can say the dots are closer to the central point, to the mark. That means they are accurate, but they are not closer to each other. That means they are not precise. In third example, in third figure, you can see they are far from the central point. That means they are not accurate. On the other hand, they are not closer to each other. I mean, they are not reproducing the same results. That means they are far different from each other as well. That means they are not either they are not accurate and they are not precise. And in the last picture, you see they are closer to the accurate point. So they are at center. That is the mark. They are on or closer to the central point. That means they are accurate. And on the second hand, the different shots are closer to each other as well. That means they are showing the repeated results. That means they are precise too. So in this example, they are precise and accurate. In this example, they are not precise and not accurate. In this point, they are closer to the central point, I mean target point. So they are accurate, but they are not precise because they are not producing the same kind of results. They are different from each other. And in this example, they are producing the same kind of result, but they are far. They are precise, but they are not accurate. So this is how you can easily understand the concept of the true value, which is the value of population, the accuracy and the precision. I hope you easily understand it. Now, we talk about the errors. What kind of errors we generally deal with when when we perform sampling? So there are two kinds of errors we generally you know deal with. Number one, sampling error. Number two, non-sampling error. So it is clear from their names. Sampling error means if there is some problem in the sampling. I mean, you didn't, there was something unclear in your sample, or your sample was not appropriate, that's why you didn't get the appropriate results from your sample. In that case, sampling error would occur. The difference between the statistic and the parameter. Okay, ji. Sampling error, basically this is the sample result and this is the true value, यानी कि population का result है ये. जितना इनके दरमियान difference कम होगा उतना sampling error कम होगा. अगर जो आपने information sample से ली है वो और population की actual information के दरमियान कम difference है तो इसका मतलब है sampling error कम है और sampling error कम होना चाहिए. सैम्पलिंग एरर किस वजह से आता है? अगर आपने सैम्पल सही तरह से जनरेट नहीं किया, दैट मीन्स योर सैम्पल इज नॉट अप्रोप्रिएटली रिप्रेजेंटिंग योर पॉपुलेशन. इस केस में सैम्पलिंग एरर अकर कर सकता है. नाउ व्हाट इज नॉन सैम्पलिंग एरर? नॉन सैम्पलिंग एरर ऐसा एरर होता है जो टेक्निकल एरर नहीं होता. दिस इज कॉल्ड ह्यूमन एरर. ये किसी ह्यूमन नेग्लिजेंस की वजह से आता है और इसकी क्या किन वजूहात से आ सकता है? उसमें नंबर 1 है पुअर सैम्पलिंग मेथड. हो सकता है आपने सैम्पलिंग के लिए अप्रोप्रिएट मेथड सेलेक्ट ना किया हो, जिसकी वजह से जो आपके पास सैम्पल आया वो ठीक नहीं है, पॉपुलेशन को ठीक रिप्रेजेंट नहीं कर रहा, तो दैट विल बी नॉन सैम्पलिंग एरर. नंबर 2 है क्वेश्चनियर. हो सकता है आपका क्वेश्चनियर ठीक ना बना हो जो आपने फिल करवाया है, जिससे आपने सैम्पल कलेक्ट किया है, उसकी वजह से एरर आ रहा है. मेजरमेंट एरर हो सकता है जो आपने वेरिएबल्स जनरेट किए हैं उनकी जो आपने स्केलिंग वगैरह किया वो ठीक नहीं थी और उसकी वजह से एरर आ रहा है. नंबर 3 है बिहेवियरल एरर. आपने कोई मिस्टेक कर दिया है, रिटन मिस्टेक कर दिया. आपने 34 लिखना था, आपने 53 लिख दिया है. सो न्यूमेरिकल मिस्टेक हो गई है या कोई इस तरह की मिस्टेक हो गई है जो आइडेंटिफाइड नहीं थी लेकिन वो आपके रिजल्ट्स को अफेक्ट कर रही है. सो दिस काइंड ऑफ एरर, these kinds of errors called non-sampling error. So in today's lecture we have talked about some basic concepts of sampling that includes the true value, the accuracy and the precision, and further we have discussed how many errors we generally deal with when we perform sampling. I hope आपको आज का lecture understand हो गया होगा. Thank you so much. Allah Hafiz.